that's not right. Let's try that again. Oh, sorry about that. That was a little bit better. Technical difficulties. So we're talking about muscles, and muscles can be three types. They are three types of, well, muscle tissue. We've got skeletal muscle, cardiac muscle, and smooth muscle. Now, most of what we're going to be talking about is going to be skeletal muscle. We will talk about cardiac and smooth muscle towards the end. So skeletal muscle helps produce movement, maintains posture and body position so we can stand upright, basically. It supports soft tissues, guards body entrances and exits. Think your throat, the different sphincters that go from different parts of your digestive tract, for example. Maintaining body temperature. About 85% of the heat that's produced to maintain your body temperature is produced by your muscles. Storing nutrients are the main functions. So we've got three layers of connective tissue associated with these, also kind of holding everything together. We've got the epimysium, perimysium, and the endomysium. This one right here, we're going to start on the outside with the epimysium. The epimysium is a layer of collagen fibers that surrounds the muscle. It's connected to the deep fascia that sort of holds all of the organs and all the other tissues in place. And it separates the muscles from the surrounding tissues. And so if you look at this picture right here, it is the lining of the individual muscle itself. And so it's going to sit here on the outside. Then moving inwards, we have the perimysium. The perimysium surrounds the actual muscle fibers in the muscle, and these fibers are known as fascicles. Forgot a C in there. So the perimysium is going to surround the fascicles, the muscle fibers. It contains collagen fibers, elastic fibers, blood vessels, and nerves. And then moving farther inward, we have the endomysium, and that surrounds the individual muscle cells. These contain capillary networks, myosatellite cells that repair damage, and nerve fibers. So when we're going outside is epimysium, that surrounds the muscle. Perimysium surrounds the fascicles, which are the bundles of muscle fibers, and the endomysium surrounds the individual muscle fibers. And then associated with this, we've got collagen fibers of those three layers can come together in different structures, typically at the end of the muscles. They can be tendons, where they come together in a bundle like this, or they can be flattened, almost like a sheet, and that's aponeurosis. And these structures are primarily responsible for attaching the muscle, which is this part right here, to the bone, which is this part right here. Skeletal muscles have a number of different structural characteristics. One is a vascular network that brings oxygen and nutrients and also removes metabolic waste, because these tissues are very, very active tissues. They also contract only when stimulated by the central nervous system, so that means we call these voluntary muscles. So like I'm moving my fingers right now, I'm doing it voluntarily. Your heart is involuntary. That muscle tissue, you don't consciously control it, which is good, because then you would forget to make your heart beat. The diaphragm usually works subconsciously, but you can consciously control your breathing rate. And so if you've ever thought about calming down and stuff like that and forcing yourself to slow your breathing, that's conscious. But for the most part, you don't have to think about you don't have to think about your breathing. You may see skeletal muscles referred to as striated muscles because they have striations, and that's just a fancy word for saying stripes. And that has to do with the types of proteins and how they're arranged, which we'll get into in just a little bit. All right, so some of the structures of the actual muscle cells. So muscle cells are going to be kind of structurally unique in that they're really long, like ropes, instead of round like what we typically think of, or sort of roundish shaped. So they have a plasma membrane, but it's got a special name called the sarcolemma, which is right here. And you can zoom in. So it's this structure right here that surrounds all these individual structures. So this is still an individual cell that surrounds it. The sarcolemma surrounds something called the sarcoplasmic reticulum, which is this blue thing right here. And what we're going to look at in a little while is voltage changes in the membrane potential is what we're going to call it, but it's essentially voltage, like electricity, is going to cause muscle contractions. We have structures called transverse tubules. So transverse tubules, if your muscle fiber is running long like this, your transverse tubules are going to be perpendicular to that. And so that's these yellow structures right here, these rings periodically. And these are going to serve to transmit action potentials from the sarcolemma, that plasma membrane of the muscle cell, into the cell's interior, which is then going to initiate the contraction response. We have the sarcoplasmic reticulum, which is that this blue structure that I told you about. It surrounds each myofibril. Now let me back up just a second. So your muscle cell is made up of smaller structures called myofibrils, and myofibrils are individual strands. And so if we were to think of muscles as ropes, the individual thread is the myofibril. If you bundle those together, you get the string. If you bundle many strings together, you get rope. If you get many ropes together, then you can kind of think of it as cables, and so on and so forth. And so you have myofibrils, groups of myofibrils together. Inside of the sarcolemma is the muscle fiber. Groups of muscle fibers are fascicles. Groups of fascicles make up the muscle. Okay, so sarcoplasmic reticulum. It's similar to the smooth endoplasmic reticulum. So we've got transport involved. And it's specialized in storing and releasing calcium ions, which are going to be really important in a little while. And we had mentioned in bones that calcium is really, really important for different physiological processes, and this, well, I guess, right, this is one of them. Myofibrils, what else did I didn't say? They're made up of protein filaments called myofilaments. And there's two types of filaments. There's thin filaments, which are the red lines down here at the bottom. If you kind of zoom in, if we zoom in a little bit, the red lines right here are the thin filaments because they're kind of thin. And then you've got the thick filaments, which are the darker colored ones. Those are primarily a protein known as myosin. The thin filaments are a protein primarily known as actin, and those are going to be super important when we're going to be talking about muscle contractions. So what we can do is we can divide up a myofilament into functional units called sarcomeres. So a sarcomere is the functional unit of the muscle fiber. And so basically it goes from something called the Z line, and it's called that because it's zigzaggy, kind of like a Z, to Z line. And then you've got other bands in there and other proteins that are going on, like the I band, which is the lighter bands right here, and the A bands, which are the dark bands right here. And this is where we get those striations that we see in striated muscle. If we stack up all of these, all the A bands of the myofibrils give us those striations. Okay, so if really to kind of get it to scale, there's millions and millions and millions of these things in your muscles to give us those colors, patterns. So this is kind of a recap of sort of what I was talking about. So you have your sarcomere, which is a section of your myofibril. Okay, you put a bunch of sarcomeres stacked together, you wrap them by a sarcoplasmic reticulum, that is your myofibril. You get a bunch of myofibrils together, surrounded by the endomysium, that's your muscle fiber. You take a bunch of muscle fibers together, and then you wrap them up in the perimysium, that's your muscle fascicle. And then you take a bunch of fascicles and you bind them together with the epimysium, and that is your skeletal muscle. And so there's these layers upon layers upon layers. All right, so a couple of things about the different filaments, structurally speaking. Your thin filaments, the red parts, basically, are made up of mostly actin. That's a sort of licorice-looking color. And then these other green, they look like springs, called titin. And those are going to act like springs, tension rods, whatever. They're going to help the sarcomeres go back to resting state once the contraction phase is over. And we'll get into that here in just a little bit. They also have a protein called tropomyosin. So it's kind of hard to see, but those are these green licorice proteins that weave all through here. That's the tropomyosin. And those are structural proteins that are going to hold the actin in place and expose the active sites. And then there's another protein called troponin right here that kind of acts like a blocker to let the muscles rest. The thin filaments, well, okay, so we just talked about that. That's fine. All right, so the thick filaments are mostly myosin, and the myosin have these heads that kind of look like golf clubs, basically, with a tail that binds to the other myosins, making these strands. So here is your tails right in here, and then your heads, and it zooms in for us so we can see those structures. Okay, so this gets us to what's known as the sliding filament model or theory, right? So we have these different filaments, the thin and the thick filaments. And so basically what's going to happen is during a contraction, these bands are going to slide past each other, okay? And so during a muscle contraction, your thin filaments are going to get pulled closer together and creating this zone of overlap right here. The A bands don't move, they stay constant. They're going to pull the I bands, those red parts, the thin filaments, together, and it's going to shrink your myofibril, and then that's going to lead to your muscle contraction. Okay, it's a little bit more complicated than that, but that's the basis of the sliding filament. We've got these different filaments that are sliding past each other, and as they come together, that's a contraction. Okay, so nerves and muscles work really, really closely together with each other, in part because they have what are known as excitable membranes, okay? So excitable membranes have repolarization and depolarization events. So we basically have changes in voltage across the membrane, right? So it becomes more positive, less positive, that sort of thing. And these changes create something called action potentials. And these are electrical impulses that we think of when nerves are like firing and stuff like that. These are called action potentials. Skeletal muscles, the ones that we've been talking about, contract because they're being stimulated by motor neurons, okay? And so where the neuron meets the muscle, that's known as the neuromuscular junction. And a synapse, we'll talk about those when we get into the nervous system a lot more. A synapse is just where two nerve sections meet, okay? Two different cells meet. So the NMJ, the neuromuscular junction, is the meeting point between the end of a neuron and the beginning of a muscle fiber. And what ends up happening is the axon from the neuron releases a neurotransmitter into something called a synaptic cleft. That's just that space. The neurotransmitter is acetylcholine that binds to special proteins that let sodium in, which changes the voltage across that membrane and starts an action potential. Here is a video clip that illustrates that a little bit more slowly and gets us started with this muscle contraction process.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so once we have started the action potential from the neuromuscular junction, now we started another process called excitation-contraction coupling. Okay, and so basically what ends up happening is the action potential travels down the T tubules that we talked about that wrap around the individual myofibrils. That releases calcium. Calcium is going to bind to troponin and change its shape, which is going to cause troponin and tropomyosin. They're a complex together to move. All right, their position is going to change. So that's going to open up active sites on the actin of the thin filament. That's going to initiate the contraction cycle. Now, here's an animation right here that we're going to watch that goes through this particular process. So this is a sort of intermediate step in the entire contraction process.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma, and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced in foldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, so once we have sent the electrical signal down the neuron, we have moved into the muscle cell, and we have started that contraction cycle that I just mentioned. Now we get into the actual cycle. So the cycle begins. We have active site exposure, that's the beginning of it. We have the formation of cross bridges. Myosin is going to bind to actin. We're going to have what's known as the power stroke as the myosin head pivots, then the cross bridge is going to detach, reactivating the myosin, and then this process will repeat as often as necessary. And so there's actually an animation that I have. The cross bridge cycle and the actual muscle contraction.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the crossbridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Crossbridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
Okay, so that's basically how muscles work. Now, what's cool about that is that took a long time to talk about it, but like right now I'm talking, I'm using muscles, and you're gesturing with my hands, and I'm using muscles. And so all of that process that we just talked about happens super duper fast. So I don't know, I always think that's pretty cool. So one of the things that we can talk about when we're talking about muscle movement is this idea of muscle tension and the generation of tension. Muscle tension, when we're talking about that, is basically pull. So when muscles contract, they create tension, that's that pull, right? In order for movement to take place, tension has to overcome the load, what the resistance is. So for example, I've got this remote control, right? If I want to move this remote control, the tension in my arm has to overcome the resistance of the load, which is basically the weight, right? And so if I want to move it, my tension has to overcome that load, is basically how it works. The entire muscle then is going to shorten at the same rate. You don't have sections that move at different times and different rates and stuff like that. And that's because all of the sarcomeres are going to contract together. Now, how fast it shortens, how fast you move it is going to depend on the cyclic rate. That's the number of power strokes per second in that contraction rate. How long the contraction lasts is going to depend on how long the duration of the neural stimulus. How long are you telling your muscles to do that? So if I'm holding this up, right now I'm telling my muscles to contract and hold this up here, and they will continue to hold this until my brain tells them to stop, or stops telling them to do so, I think is a more accurate description. If there are free calcium ions in the cytosol, now cytosol is just the fluid inside of the cell, just sort of floating around. So if there is calcium then floating around, then the contraction will continue. ATP, if it's available, the contraction will continue. If it's not, it stops, because the ATP is required to break the cross bridges, right? So once the ATP goes away, you can't break those cross bridges. Let's see. So calcium ions, as they get pumped back into the sarcoplasmic reticulum, so out of the cytosol, out of the sort of free floating in the cell, the calcium, because the concentration is going to be going down, it's going to detach from the troponin, which is going to cause that tropomyosin complex to reset to its original position, and that's going to hide those active sites. And so then the myosin heads are not able to form those cross bridges, which is going to stop the contraction. So then, like, if I want to stop the contraction, I'm not able to create that tension. An extreme form of this is rigor mortis. Now, rigor mortis is fixed muscle contraction. So remember, you have to have ATP to break the cross bridges. So if you have no ATP, you don't break the cross bridges. You also don't have the ion pumps moving stuff in and out to stop those. And so basically what ends up happening is calcium builds up in the cytosol, keeping the cross bridges engaged. Because the active sites are open, that's the calcium part, and the cross bridges stay engaged, that's the ATP part. You know what, ladies and gentlemen, I think I'll call it there. That's a lot of information to go through. So go ahead and click over here in just a second and watch the next video. Still talking about skeletal muscles, but now we're going to be talking about using them. We'll hit on some stuff like how exercise is important, what happens when you get older, and stuff like that. So if you have any questions, send me a remind message, send me an email. Otherwise, I look forward to seeing you all again soon.